This is a leaf, and this. So is this. Leaves come in a multitude of different shapes, colors, and sizes, but they all share a critical function, and that is photosynthesis. To maximize photosynthesis, they share some common traits. In this case, they share a flattened lamina to maximize surface area for capturing light, and they generally share a similar shade of green. But beyond that, the leaves are the site where the majority of transpiration takes place. To understand how a leaf properly functions, we need to look at it at a cellular level. While we can figure out the form and function of leaves at the macro level, we can do the same at the micro level on the form and function of cells. On this slide is a Doryphora sassafras leaf outlining the basic cellular structure of a leaf, the top being the surface of the leaf and the bottom being the underside. In this particular slide, cellulose stains blue and lignin stains red. You can see the vascular bundle here, with the xylem here staining red because of the lignin in the cell walls, and the phloem here, with their companion cells. The xylem carry water for the leaves for photosynthesis, and the phloem can carry the photosynthate, or the sugars created by photosynthesis, to other areas of the plant. If we look along the surface of the leaf, we can see that there are cells that are packed tightly and in a very orderly way. This section, which is about 3 cells thick, is the palisade mesophyll. These cells are filled with chloroplasts, which give many leaves their green color, and being on the surface, the palisade cells are capturing all the light that is coming and hitting the top of the leaf. Underneath the palisade mesophyll is another layer. Here you can see that there are a lot of empty spaces between the cells. This layer is called the spongy mesophyll. This area allows for gaseous exchange, owing to all the empty space. It is in the spongy mesophyll where carbon dioxide is taken in and oxygen and water are diffused from tiny pores called stomata on the underside of the leaves. If we zoom in, we can see the stomata on the underside of the leaves. The stomata have 2 guard cells that can open and close to allow carbon dioxide in and unavoidably water out, as part of the photosynthesis transpiration compromise. You can see that here and here. So, to recap, leaves are the primary photosynthetic organs in plants. At a cellular level, there are 2 primary layers. On the top of the leaf, you have the palisade mesophyll, which is responsible for the capture of light. And then, on the bottom of the leaf, you have the spongy mesophyll, which is where the exchange of carbon dioxide, oxygen, and water takes place. Also on the bottom of the leaf, you have the stomates and the guard cells, which control transpiration of water from the leaf alongside the uptake of carbon dioxide and oxygen. Finally, you have the vascular bundles, which are the xylem and phloem. Xylem for water and phloem for sugars.